Yes.
Now you can start, Raúl.
Yes, I will introduce you. Okay.
Great.
Um, Good afternoon, everyone. Um, Today we have the pleasure to introduce Professor Gerd Kullak-Ublick from Zurich, Switzerland. Um, he is full professor of pharmacology and toxicology, and his background also includes gastroenterology and hepatology uh, at the University of Zurich for many years. And he's interested in research in many topics that are pertaining to drug-induced liver injury. For example, um, assessing drug-induced tissue injury in vitro, cholestatic liver diseases, pharmacogenetics and genomics in liver diseases, safety biomarkers, gene regulation by nuclear receptors, and bile transport and metabolism. Raising efforts in jointly efforts in different European academic centers, but also the industry, have allowed to launch the TransBioline project because of the TransBioline consortium, aim to identify novel liver safety biomarkers for drug-induced liver injury. This is a topic that in which Gerd Kullak had been very much involved. So his lecture today is the novel biomarkers for the assessment of drug-induced liver injury, both in the diagnostic and the prognostic sides. Thank you very much for joining us, and you can start.
Thank you very much, Raul, for the nice introduction. And it's a big pleasure to be able to present some of our data on biomarkers at this conference today. So just by way of introduction, drug-induced liver injury is defined by biochemical criteria. So by international consensus, an elevation of the two transaminases, ALT or AST, to fivefold or more than fivefold the upper limit of normal is considered a sign of possible DILI. An elevation of alkaline phosphatase to twofold, or the combination of ALT or AST threefold plus an elevation of bilirubin twofold. We'll be coming to that just a little later. This is what we call the biochemical criteria for Hy's law, which is a potential sign of severe liver injury. So in drug development, so in clinical trials, phase 2, phase 3 studies, there are recommendations from regulators how one should handle elevations in liver tests during the clinical trial. And obviously the idea behind this is to prevent serious liver damage from happening within a clinical trial. So if a patient has an increase of ALT or AST to eightfold the upper limit of normal, then normally interrupting the study treatment is recommended. Alternatively, an elevation to fivefold for at least 2 weeks would also trigger interrupting treatment. And again, this constellation ALT, AST more than threefold plus a bilirubin of twofold, or in very conservative terms, the combination of ALT, AST threefold together with an INR of more than 1.5. This, in FDA terminology, would also warrant interrupting treatment. Again, clinical symptoms of liver injury, which are obviously less specific in combination with transaminase elevations, would also lead to possible interruption of drug treatment. Now, ALT and AST are very sensitive and widely used markers of liver injury. What they do not do is to predict the clinical outcome. So you can have a very high elevation of ALT and still recover within a short period of time. So there is little predictive value in ALT and AST. And for that reason, these transaminases are not part of any of the known prognostic models, such as the Child-Pugh score, MELT, or the King's College criteria for liver transplantation. So in fact, what we're doing is basing decisions on whether or not to stop a drug on markers that do not actually reflect the outcome or even the severity of liver injury. And that's really the reason behind these initiatives that aim to identify new biomarkers that may be more prognostic or more specific to identify patients at risk once liver injury has been identified. Now, generally, all new lab tests, biomarkers, should be easy to measure. There's little point in having highly specialized analytical setups, which are available in 2 labs in the world. These should be readily available in routine labs. And the diagnostic yield should obviously be better than ALT or AST because we already have standard lab parameters in all routine labs. So there is a categorization of biomarkers according to the so-called BEST categorization. This is an FDA NIH categorization. BEST stands for biomarkers, endpoints, and other tools. And you can see these lists of 7 different categories of biomarkers: diagnostic, monitoring biomarkers, pharmacodynamic or response biomarkers, predictive biomarkers, prognostic, safety biomarkers, or susceptibility or risk biomarkers. So all of these have different sets of data that are required in order to qualify a new lab value as a biomarker. And what we've really been focusing on in our new drug-induced liver injury biomarkers is safety and prognostic biomarkers. So that's really what we've been studying. And different regulators have different regulatory processes. You can see the FDA process on the right and the European, the EMA process on the left. Essentially, obviously, the common ground here is you initially submit a plan of what you intend to do, a letter of intent. Then you have to generate the data to basically justify your initial intention. And then you have, at the end of the enterprise, you have the full set of data for full qualification. So in TransBioline, we're somewhere between the light gray and dark gray stage. So we're in the process, for at least for some of the organ work packages, of aiming for full qualification of new biomarkers. So the TransBioline Consortium was a public-private partnership funded by the IMI, the Innovative Medicines Initiative, which is actually the biggest public-private partnership platform globally. And IMI is nowadays called IHI, Innovative Health Initiative, which is just the successor of IMI. And within TransBioline, we had several academic partners, as shown in this cartoon top left. We had small and medium enterprises shown top right, pharmaceutical companies shown bottom left, scientific academic experts, and also representatives from regulators and ethics experts. So quite a large consortium with 27 partners and 5 different organ work packages. Liver injury was one of the work packages. We also had organ toxicities for kidney, pancreas, vascular injury, and central nervous system injury. All of these were different work packages within the consortium. So the centers that were involved in the DILI work package are shown here on the bottom right. We had the universities of Nottingham, Zurich, Munich, Malaga, Reykjavik, Gothenburg, and Bern contributing samples. So a widely distributed network. And the consortium had 2 phases: an initial learning phase, which aimed to recruit 100 DILI patients, and a subsequent confirmatory phase that aimed to recruit 200 DILI patients. these patients were reviewed by an adjudication committee. So every single DILI case that was identified was reviewed by a group of experts in drug-induced liver injury to really assess whether these cases were true DILI or possibly liver injury caused by non-drug causes. And this is what you can see in the bottom right panel, so-called non-DILI acute liver injury cases. These were the cases that were taken out of the DILI category because other confounding causes were considered to be causes of liver injury rather than a drug-induced cause. So we had a total of 271 DILI cases, 100 in the learning phase and 171 in the confirmatory phase. And the biomarkers that we were interested in are shown here. You can see the traditional standard biomarkers shown in brown in the middle of the slide: ALT, AST, INR, and bilirubin. And on the left, you can see the new biomarkers that were of interest: HMGB1, which is a transcription factor and essentially a marker of cell necrosis. We had keratin 18, which is a cytoskeletal protein expressed in hepatocytes. About 5% of hepatocyte protein are made up of keratin 18. There are two forms, a full-length form and a caspase-cleaved form, CCK18, which is a marker of apoptosis. On the far left of the slide, you can see a microRNA, mRNA 122, which is a liver-specific microRNA, and then also a marker which is already quite well known, a mitochondrial marker, GLDH, glutamate dehydrogenase, which has been around in routine labs for 20 or so years, but only few routine labs tend to measure GLDH, and so we included this as a safety biomarker in Transbioline. On the far right of the slide, you can see immune markers, MCSFR1, which is a macrophage marker, and increased levels of MCSFR1 are considered to reflect immune activation, as well as osteopontin, a cytokine, again, also a marker of immune activation. So these were the new biomarkers under study. And just to very briefly elaborate on one other diagnostic test that was looked at, this is a patient-specific causality assay called MetaHeps. So what happens is when a patient has suspected drug-induced liver injury, peripheral blood mononuclear cells, PBMCs, are isolated and monocytes are plated in the lab, and these monocytes are then subsequently converted to a monocyte type cell called MetaHeps in a 2-week incubation procedure, and these MetaHep cells are then subsequently exposed to the drugs, which we suspect caused DILI in the patient. So on the example on the right, you can see a patient who had been taking diclofenac, shown in red, tadalafil, and ciprofloxacin, and you can see that the signal was positive for diclofenac, but negative for tadalafil and ciprofloxacin, indicating that diclofenac was the presumed cause of DILI in this patient. So especially when several drugs have been taken at the same time, immune-mediated DILI can be identified by this test. And just one example of that, a patient taking several drugs, including an immune checkpoint inhibitor, as well as metimizole, pantoprazole, ibuprofen. In this patient, pembrolizumab was identified as the DILI-causing drug by the MetaHeps test, whereas another different patient shown on the right, this patient also took pembrolizumab, but also a herbal called imodin, and in this patient, imodin was identified as the DILI-causing drug, so herbal-induced liver injury, whereas pembrolizumab was negative in the MetaHeps test. Okay, so just moving on. So these were the different patient groups that were studied in Transbioline. We had essentially 5 groups of patients in the liver work package: true DILI patients, drug-induced liver injury, with the biochemical criteria discussed earlier. Then these patients in whom a drug cause was excluded, but who still had acute liver injury, of some other cause. These were the so-called non-DILI acute liver injury patients. Then we had a group with either alcohol or non-alcoholic fatty liver disease, the NASH or NAFLD group. Then a group of patients who had chronic inflammatory conditions who were being treated with immunosuppressants such as methotrexate. And then finally, healthy volunteers as a comparator group on the right. The assays were performed by a biomarker startup company in Reutlingen, Germany, Steatotic, with two different platforms, an immunoaffinity LCMS platform for osteopontin, GLDH, MCSF1R, and HMGB1, as well as an ELISA for K18 and CCK18. Okay, so as always with regulatory documents, you need to define a context of use within which you plan to use this biomarker, should it be qualified by regulators. So where is the added value compared to ALT and AST? And the context of use that we defined was based on a drug development use. So patients who fulfill the official criteria of DILI, shown in the top right panel here, should be further characterized by use of these new biomarkers to see whether the biomarker in question is above or below a certain threshold. So there's like a second layer of stratification to identify how much at risk the patient is once these traditional conservative criteria are fulfilled. And so if the biomarker is below what we would call the DILI threshold, you could justify maintaining or restarting the investigational drug despite having these traditional criteria fulfilled. Whereas if the biomarker threshold is exceeded, so if the biomarker value is also above this threshold, then you proceed with the current recommendation to reduce or interrupt dosing and wait for things to normalize. So it's like a second. The idea here would be to possibly prevent premature termination of a potentially useful or even life-saving drug based on the levels of the new biomarker. Good. So in the data I will show you now are based on the first 100 patients. This is the DILI learning set. We do not yet have the final analysis of the full DILI set, the full 270 patients, but we have the first 100 patients analyzed. And you can see that distribution here between different phenotypes, as expected. Hepatocellular DILI with predominant ALT elevation is the biggest group here. Cholestatic patients with predominant ALP elevation are a smaller group, and we also have mixed type injury. And then we had about 44 and 43% of patients in the mild or moderate severity category, and 13% with severe DILI at presentation. Outcomes were progression to severe DILI in 15 cases, so 15% progressed to severe DILI. In other words, from the time point of initial identification to the time point of final assessment, there was a progression or worsening in DILI. 12 of these 15 actually had transplant or fatal outcomes, and 3 had unknown outcomes. And just for comparison, the non-DILI, so these are the acute liver injury patients without a drug cause, qualitatively a similar picture. This is a smaller group of patients, 40, again, mainly hepatocellular, mainly mild or moderate severity at presentation, and again, a relevant number of progressors also underwent transplantation. So these are the first biomarker results. Now, each of these box plots has the same set of patients. You can see at the bottom, DILI, drug-induced liver injury patients, then the acute liver injury of non-drug cause, non-DILI, then the NAFLD or alcoholic fatty liver disease group, the rheumatoid arthritis or psoriasis group, usually patients taking methotrexate, and then the healthy volunteer group. And you can see the comparison between the groups here for the different biomarkers. So we have GLDH at the top. GLDH1 is protein concentration, and GLDH2 is GLDH enzyme activity, highest in DILI and non-DILI. Keratin 18, again, highest in DILI and non-DILI compared to, obviously, healthy volunteers, which were much lower. Caspase-cleaved keratin 18 was, again, highest in DILI and non-DILI. So you can see across all the biomarkers, DILI and non-DILI were very similar, with alcoholic fatty liver disease having elevated levels, but less than the DILI and non-DILI groups. And this also goes for the biomarkers at the bottom, osteopontin, MCSF-R1, and ALT, interestingly, as well. Okay, so just moving on to the next slide here. So this is now zooming in a little bit just on DILI and non-DILI compared to healthy volunteers. So we're leaving out the alcoholic and NASH groups and the rheumatoid arthritis groups. These are now just DILI, non-DILI, and healthy volunteers. And again, you can see the results are very similar for DILI and non-DILI. And as expected, these biomarkers are all higher in the liver injury group compared to the healthy volunteer group. So no big surprise here. Now, interesting is the comparison between DILI and acute liver injury of non-drug causes. You can see at first sight, the results look very similar. If you look at the panel on the top right, this is CCK18. The non-DILI group tended to have slightly higher levels compared to the DILI group. And if you look at the the bottom second from right panel, MCSF1R, again, the non-DILI group tended to have slightly higher levels compared to DILI, but these results are not significant and should be seen with caution. And just the ROC values for that previous slide, DILI compared to non-DILI, you can see that none of these biomarkers was really able to differentiate between DILI and other causes of acute liver injury. If you look at the AUC values here in the left part of the table. So the next question was, what about progression? So these are patients who, from the time point of initial diagnosis of DILI, progressed to more severe DILI, all the way up to either a fatal outcome or liver transplantation. And you can see the legend here, yes, stands for yes, progressed to more severe liver injury versus no progression to more severe liver injury. And what you can see is that the DILI patients who progressed tended to have higher HMGB1 levels, shown on the bottom left. They also had higher osteopontin just next to it and higher MCSF1R levels. So these three biomarkers seem to be a prognostic value, especially MCSF1R and HMGB1. So that was interesting. And just moving to the next slide. So just to remind you, the number of patients we're talking about here, if you look at the top right panel, of those patients who were mild at presentation, 44 cases with mild DILI at presentation. Of those 44, 2 progressed to moderate, 1 progressed to severe, and 2 progressed to either transplant or fatal outcome. Of the patients who were moderate at presentation, 43, 4 progressed to transplantation or fatal outcome. And of the patients who were already severe at presentation, 6 progressed to transplantation or fatal outcome. And yeah, this is just the bottom left panel just shows the progressors according to DILI phenotype, and you can see in all 3 groups we had about the same proportion of progressors, so no clear pattern, hepatocellular versus cholestatic versus mixed injury. Now, looking at the entire 100 patients, if you look at the prognostic performance of the biomarkers, progressors versus non-progressors, you can see, at least for HMGB1, an AUC value of more than 0.75, which is a significant result, indicating that HMGB1 was able to distinguish between progressors versus non-progressors. And MCSF-R1 was close with 0.72, K18, keratin 18 was 0.69, and osteopontin was 0.68. So these three are promising, and MCSF-R1 was significant. HMGB1 was significant for differentiating progressors from non-progressors. MCSF-R1 was close. Now, if we zoom in on only those cases who had mild injury at presentation, so these were the 44 cases with mild liver injury at presentation. Now, the reason we looked at these was because in a drug development setting, where patients generally have normal liver function at the outset, by far the most common type of liver injury that is seen is mild liver injury. Grade 2, grade 3 elevations of ALT, AST, whereas severe liver injury is a rare finding. So these are the more common patients that are seen. And obviously in that context, it would be particularly interesting to see if any biomarkers can differentiate between progressors and non-progressors. Again, yes indicates progressors from initially mild to more severe liver injury versus non-progressors, labeled with the no. And there were trends. So you could see, if you look at HMGB1, bottom left, you can see there was a trend for progressors to have higher levels. Same is true for MCSF-R1, K18, and CCK18. So qualitatively a similar picture compared to the entire DILI cohort, but obviously probably slightly less statistically significant, which is also based on the cohort size, with a total of 44 patients in this mild DILI cohort. Again, the ROC values for that group, these mild injury cases at presentation, of all the markers, essentially only HMGB1 showed a significant value here, as indeed did bilirubin with 0.79, which is part of the Hy's law algorithm. So bilirubin actually still scores best in this table, and MCSF1R came close to the threshold with 0.68. So just looking at the, this is what we call a Pearson correlation. So this essentially correlates different biomarkers with each other. And what you can see is that, not surprisingly, full-length keratin 18 correlates with caspase-cleaved keratin 18, which is an expected finding. And similarly, GLDH activity correlates with GLDH protein concentration. So these were kind of expected findings. Possibly more interesting, this box here shows that the patients, these are non-progressors, and you can see that keratin 18 and GLDH activity show a mild correlation. See these light red dots in this panel here. This is the correlation between keratin 18 and GLDH activity. And and then we also had ALT and GLDH in progressors, which is actually this box here shown in, which is framed in light blue at the bottom right. If you look at the blue or turquoise data points in this panel, you can see a trend towards a correlation here between ALT and GLDH. So, but no exciting biomarker combinations that would increase the diagnostic yield compared to individual biomarkers. So in summary then, biomarkers are an essential tool for drug development. potentially contributing to patient safety. The best possible way of assessing new biomarkers is in collaborations like we had in TransBioline, academic partners with pharmaceutical companies and SMEs. So the public-private partnerships have I think, the strongest potential to bring together different stakeholders who can contribute each according to their potential and expertise. We know that DILI is a leading cause of drug withdrawals post-approval and a leading cause of drug attrition in development. So there is an urgent need to develop new biomarkers that help us assess these patients in clinical trials. And the current standard lab parameters tend to be sensitive, but they lack specificity, and they also lack the prognostic value, with the possible exception of bilirubin. We have encouraging results for some of these biomarkers as prognostic biomarkers, notably MCSF1R, HNGB1, and osteopontin. MCSF1R and osteopontin are both markers of immune activation, and this does sort of confirm the immune component as being very key in the pathogenesis of idiosyncratic or severe liver injury. So idiosyncratic, which is another way of saying immune-mediated liver injury, is a very important pathogenetic mechanism behind liver injury. And yeah, we obviously hope to continue the qualification of these biomarkers. There is a consortium at the Critical Path Institute in Arizona, the PSTC, Predictive Safety Testing Consortium, which is continuing the regulatory efforts that were started in TransBioline, so that we hopefully will be able to fully qualify some of these biomarkers with FDA during the next 12 months or so. So thank you very much. That was all I had to show for today for new DILI biomarkers.
Thank you very much, Gerd, for this very interesting and very live presentation, because the data is not yet published. So we appreciate your sharing this very interesting information. Is there any questions from the audience? Because indeed, as you said, we need really novel biomarkers in DILI, because otherwise, as it's a diagnosis of exclusion, it takes a lot of effort, analysis that costs a lot of money, no? Images, or even liver biopsy, in order to other potential causes. So the need for biomarkers in DILI is really a real need. But as you have presented, it's very challenging, no? It's difficult to get something that is really liver specific or drug-induced liver specific. I don't know if there is any questions. Yes, Inbal.
Yes.
Inbal.
Hi.
Hello.
Thank you. Fascinating talk. I'm asking about something that you might have already presented and I missed. The cohort for this trial, where did the patients come from? Were they random walk-ins, or patients who were part of a clinical trial and were vetted for all the other parameters and had a bad response to whatever compound they were given?
Yeah, thanks. Good question. So these were actually real world patients in hospitals. So these were the academic hospitals that were involved. These were patients that were treated for various indications and happened to develop drug-induced liver injury on antibiotics, on cancer drugs, whatever. So it was a very heterogeneous group in that regard. The possible advantage being that the hospital patients obviously develop different levels of severity from very severe to mild injury. And so at least we had that spectrum covered, but these were normal hospital patients.
Okay. And the cell culture that so from each patient that developed a bad response, you collected the monocytes and tested for that compound?
Actually, not only from a select group, not from all patients. The reason being that particular technique was initially being done in Munich and then was transferred to Zurich. So there were some logistic issues. We had to refine the method to be able to freeze PBMCs and then be able to use frozen PBMCs, which actually was not the case at the beginning of the consortium. So we didn't have that many patients analyzed yet with that method. Yeah, but a few. We have a handful, about 20 or so, were included.
Was there any correlation between the cellular response and the patient?
Yes, absolutely. Yes. So I think the causative drug was identified in the 20 cases that we did manage to include, with maybe one or two exceptions. But I think drugs that are readily identified by the technique are antibiotics, NSAIDs, immune checkpoint inhibitors. It works well for that.
Amazing. Thank you.
Yes. Any other? Oh, I would like to ask you a question, because you have always shown interest in biomarkers validation. When we are running a clinical trial, how should what should be the trial designs or the endpoints to best support the qualification of these biomarkers? Because at the time, now we are just identifying the patients, having a centralized adjudication committee, and then making an stratification that is the randomization. But we collect blood sample in order to make those analyses later, but not in order to see the prognostic, to make a prognostic or risk stratification of the population. So what do you think?
Yeah, it's exactly you hitting the nail. So ideally, if you had a phase 3 trial in a pharmaceutical company, you would plan to measure these biomarkers throughout the study. So you would maybe take a baseline blood sample from each patient and at minimum freeze it away. And then, I don't know, if you have 1,000 patients in the trial and 26 develop DILI, then you can selectively look at those patients and maybe compare baseline values with the values that come when DILI has occurred.
Yeah.
And that would allow you to, to also compare it then to the other patients who did not develop DILI. From our experience with companies, teams are always reluctant to do things that they don't have to do. And the question that always comes is, yeah, but are these markers qualified, or are they purely experimental? And that's really why you why we have to try to get at least some of these markers qualified, because only then will companies accept the effort of including these markers. So it's a bit of a sort of catch-22.
Yes.
We actually need clinical trial samples to reach full qualification, but the companies are only willing to do that once we have full qualification. So you have to sort of hope that some will be cooperative and will be willing to measure these markers anyway. Yeah.
Yeah, but do you think we can come up with thresholds, values to this?
Right. I mean, that would be the next step. Absolutely. I mean, I think that would be actually what we had in the context of use, would be to define a threshold to really say, above the threshold, it's
Sure.
DILI, below the threshold, it's not DILI. That would be the next step then. Once the qualitative value has been accepted, I think then we would really need to see, what are the thresholds, but that really would require having clinical trial samples where patients are normal at the beginning and develop DILI during the trial, so you can really say what the delta is from beginning to end.
That needs a really collaborative effort indeed,
Yes.
and
Yeah, absolutely.
bringing together the pharmaceutical industry, or those. Yes.
Yes,
Okay.
definitely.
Thank you, Gerd. Professor Dominique Leray has the word.
Thank you, Gerd, for this night talk and a very interesting topic. And I have 3 questions. Sorry. One question is the time frame for collecting the sample is very important to have an optimized result with these markers. In other words, when to consider the collection of the samples just at the time of liver injury, or whether shortly after, finally it's less performing.
Very good question. So we had I think in most cases, the sample was collected at the time that the DILI was identified. So probably, not necessarily at the peak of DILI, but at the moment in time when elevated ALT levels were seen, that was when the blood sample was collected. With follow-up blood samples after, possibly 1 or 2 follow-ups, 1 week and 4 weeks later. With one exception, this MetaHEPS technique does not work in samples that are taken at the time of peak ALT levels, probably because the immune system is too strongly activated. And there we saw that we have to wait for things to normalize. ALT has to normalize, and maybe after 4 weeks or so, and you have to then take the blood sample. It doesn't work well at the peak of the DILI episode. But for the other biomarkers, there we really took the sample as soon as possible after identifying DILI. Whether or not that's the best point in time, that is, of course, an open question. Again, for that, clinical trial patients would be ideal because you could then sequentially plan sampling these patients, and that would help us to answer that question.
Yes, second question was related to MetaHEPS. Because MetaHEPS exists for near a decade.
Yeah.
And so at the beginning, there is a question about specificity of MetaHEPS. And my question, do you use it in clinical practice, and why agencies do not use MetaHEP in the clinical setting? Because it would be very important finally to have accurate diagnosis in clinical trials.
Yeah, it's a good point. This is partly for logistic reasons. The group which initially developed it was a small academic group in Munich, but they didn't have the resources to really test a clinical trials patients or a sufficiently large group of patients. They had 250 DILI patients from Munich. But then that group gave up with the technique, and it was transferred to Zurich halfway through the Transbioline Consortium. And so we're currently now at the stage of having submitted a letter of intent. So this is the first regulatory document to FDA. And the letter of intent came back with a long list of questions, exactly as you said, relating to specificity, the lack of drug metabolism by these cells. They say, isn't it normally the metabolite that triggers DILI rather than the parent compound? So there are a lot of open questions here, which are very justified and also difficult to answer. The funny thing is that it does seem to work to a certain degree in the way the setup is being used now, for whatever reason. So maybe there is some innate immune memory which is present in these cells, which allows the cells to react even to the parent compound. But I think we're still nowhere near the being able to answer all the questions there. So a lot of question marks. But with a bit of luck, we might be able to at least get FDA agreement, which would be the so-called letter of declaration. And with that, companies would probably be willing to at least test their DILI samples in clinical trials. And only then will we make progress, not before then.
And the first point I'd like to raise is that you are showing that for some cases who have statistical significant marker, but the question is that at the level of a single patient, since your data are showing that even when there are statistical differences, there is an overlap between DILI and non-DILI, for instance, and how to solve it out, because to have something which would work in a single patient, not for a group of patients.
Yeah, I think that comes back to Maribel's question of defining the threshold. At the end of the day, it would be used like any other standard lab value, like ALT or AST. You would have to define a normal range and what you can expect to see with liver injury. That's probably what it comes down to. Trying to get these normal ranges in a cohort of healthy volunteers compared to a relevant number of DILI patients, say 300 DILI patients compared to 400 healthy volunteers. I think with that, you could probably define ranges which make sense.
Okay, thank you.
Thank you, Dominique. Any other question to our very outstanding guest, Gerd? There are many people attending this conference from many also that have been part of the cost action priority network. Yes, I'm seeing here Ali Riza. Hello. Yes. I wanted to ask you, meantime, any other question come up, because most of the these biomarkers that you have been talking about are about necrosis, apoptosis, liver injury, but hepatocyte injury or mitochondrial biomarkers, as you said, but they're much less about cholestatic markers. So what do you think? Those biomarkers are probably the hepatocyte injury will be the most likely to translate into clinical practice. No, what do you think about?
Yeah, absolutely. The actually the cholestatic markers would be, for example, bile acids.
Yes.
And which it's actually an interesting question. To what extent are bile acids really markers of cholestasis? And we did look at bile acids as well. This was the Salamanca group.
Yes.
And what we saw was that bile acid levels were significantly elevated in DILI compared to healthy volunteers, for example. And also the non-DILI cases, as expected, also had elevated bile acids. And we did stratify, according to primary, secondary, conjugated, non-conjugated bile acids. And some results were seen that, for example, the ratio of primary to secondary bile acids, that sort of thing improved the prognostic value. And in the end, the bile acid data were incorporated into the MELT score, and that was then seen to be that was then
improve, no?
seen to, that was then seen to be. I just looked at the abstract briefly. That was then seen to be improve the MELT score. But the bile acids have been looked at for decades in the context of liver injury. This was already this goes back to Franz Stellard, many decades back, and it was never really a reliable marker for liver injury. And with one exception, intrahepatic cholestasis of pregnancy, there we know that total bile acid levels are early markers of ICP. But with the exception of that, they've never been considered to be, of diagnostic value. But possibly now with these new data we have in the Transbioline Consortium, we might be able to see patterns there. At least there were a couple of bile acids that were also seen in the previous consortium, SAFE-T, where we also had certain bile acid species that were significantly elevated in idiosyncratic DILI, even in non-cholestatic patients. So it still needs more work, but it's possible that bile acids may turn out to be more useful as markers than previously
thought
recognized. But beyond bile acids, ALP and GGT, there was nothing really striking in the Transbioline results that would suggest a prognostic value there.
Yes. But even so, what I realize is that it's very difficult to debunk MELT score.
Yeah.
It's
absolutely. Yes. It's absolutely.
As a prognostic, no? Prognostic.
Right.
Yes.
Of course.
Although it has to be said that MELT encompasses a lot of different parameters that have to be measured simultaneously, and this will be just only one or maybe a combination of different biomarkers that might be the future, no?
Exactly. That was the conclusion of the safety consortium as well, the predecessor of Transbioline, that it's in the end the final publication showed that the MELT score in combination with MCSFR1, for example, gives a more accurate prediction. But it was, as you said, in combination with the MELT score. That's maybe what it'll come down to here as well. Yes.
Well, very interesting indeed, because it's quite thought-provoking, no? All the data that you have presented.
Yeah, absolutely.
There is still a lot to get out, no?
Definitely.
So that's good. So I don't see any more questions from now. Sorry. And if there are no more questions, and Daily is here also. And you want to say? There are many. Yes, has been very well.
I think it's great progress. I think some of the assays are quite technically challenging, I would say. I think we have made progress on the susceptibility because we can look at DNA, and that's available widely these days in many hospitals. But I think we need to make some of the biomarker assays a little bit more straightforward. But I think the best ones will progress in this way because it is possible. You just have to decide what is worth progressing and what is not. And I think that's going to be one of the next steps.
Yes, in order to translate into clinic, yes, for sure. Well, Joseph Glaza has joined also, Ana Serras from Portugal, Camila from Malaga, Maria Montes, I have seen her before, Jessica Wong, so many, Gerd, you've been very well attended,
Absolutely.
from Nottingham, from many places. So we thank you really very much for your time and for preparing this very, very nice and comprehensive presentation, and for giving us the latest information on this issue. Thank you very much, Gerd.
Thank you. Thank you.
Yes, I will
Okay.
also use these reactions that I like. Okay. Thank you very much, Gerd. We keep in touch.
Absolutely. Bye-bye then.